Now, there's another type of membrane transport that we didn't discuss in those previous blue slides, and that is invasion. So the cell can be invaded by, well, invaders, I suppose, but a perfect example is chlamydia trachomatis, which is a sexually transmitted infection. But effectively, we have these elementary bodies of chlamydia. They can surround an epithelial cell of the urethra, and after about half an hour, they'll enter that cell. Within 1 to 8 hours, they'll form this inclusion. They'll start to form inclusions, and 12 to 24 hours later, that inclusion will start to show reticular bodies, reticulate bodies, and over the next 30 hours, and over the next the next 24 hours or so, that inclusion will increase in size and the number of reticulate bodies will increase, and then you'll start to have those elementary bodies reform again. After about 48 to 72 hours of total infective time, that cell is going to die and elementary bodies are going to be released out into the wild of your epithelial tissues to infect more cells. So this is the life cycle of chlamydia within a human being. So within 3 days, one cell can infect thousands of cells. So you are very infectious with chlamydia once you have contracted it. And it can be completely symptomless. So enjoy that, internet. All right, now let's talk about surface area to volume ratio now. We're going to do a little bit more math. I know. Unit 1 we did math. It's not over yet. So surface area. The surface area of a cube is going to be 6a squared if the length of the sides is a. Now, the volume of a cube is a cubed. A times a times a. Yes. A cube's volume is a cubed. That's why it's the third power is cubed. So that means that if our side length is 1, our surface area will be 6 because it's 6 times 1 squared, and our volume will be 1 because it's 1 cubed, 1. If our side length is 2, then that means that our surface area will be 24. 6 times 2 squared, which is 6 times 4, which is 24. And our volume will be 8, which is 2 times 2 times 2, which is 8, which is 2 cubed. And if our side length is 3, we get 6 times 9, which is equals 54, and 3 times, well, we're 3 cubed, which is 27. So effectively, the ratio here is 6 to 1, 3 to 1, and then 1, and then 2 to 1. So for every step increase in size, our ratio is going to get smaller. Our surface area to volume ratio is a problem if we treat a sample organism as a simple geometric shape. So these are some simple geometric shapes. Here we have a sphere. Here we have a D20, here we have a D12, here we have a D8, D6, and D4. This is the internet, I have to show my nerd cred. So eventually our volume is going to increase at a greater rate than the surface area. Since the rate of diffusion relies on surface area, we're going to find ourselves at a point where our cells are going to choke to death. I mean, they're not going to be able to diffuse properly to support their total volume. So remember, of course, that our cells are roughly spherical. They have the worst surface area to volume ratio problem. So effectively, how do we have an inside at all? Because last I checked, we are quite a bit bigger than a single cell. We are quite a bit bigger than but a single micrometer. If our cells went without oxygen for 5 minutes, many of our cells would die. Our brains would die within 30 seconds. So we can't diffuse what we need. We certainly would not have our mitochondria be able to produce ATP without oxygen, and that's first, second, and third rule of what they do. That's why they do Rule 3 at all, so, you know. Effectively, our air comes into our lungs, and the rest of our body is basically saying, well, we need air too. We need oxygen as well. So how are we going to get that? Now, our solution is going to be transport systems. This is Unit 4, but that's a foreshadowing of future videos. So I'm going to talk now about fractal branching for a moment and how it pertains to surface area to volume ratio. We keep seeing this pattern show up in nature, this fractal branching. The short answer is because of surface area to volume ratio, why we keep seeing it. Larger vessels will suffer from all the same diffusion issues as geometric shapes. So a large vessel, like this, if we're trying to have oxygen out here, diffuse to the center of this, it's going to take some time. Every single dot here is 1 second in this large vessel. If I have a small vessel like this, or like that, and many small vessels like this, eventually if I have enough of these, and I have oxygen here, then I just have a couple of seconds required to get into the center of these vessels. And I have now filled, if I have thousands of these versus one of these, suddenly it's only taken me 1 or 2 seconds to completely oxygenate all of this blood compared to the 30 seconds it took me to oxygenate all of this blood. So that's why we see this fractal branching. A number of smaller vessels is easier to diffuse substances across all of them at once compared to one large vessel. And you get the same volume. So the surface area is greater, and the diffusion is reliant on surface area. The volume is the same because you have the same cross-sectional area with thousands of vessels versus one large vessel. Another issue that you run into is the rate of flow. If your flow rate is really fast through these large vessels, because you have a large volume moving through one vessel, you have a lot of blood flowing through a lot of individual vessels here. That will slow down your blood flow. So you have the same flow through these large vessels here as you have through all of these individual small vessels here. The total cross-sectional area is greater, but the flow through these vessels must be the same as all of these endpoints. Because out the other end, the outcome has to be the same. You have to get out what you put in. So if your large vessel has a certain amount of blood flowing, a certain volume of blood flowing through it, and that leads to a whole bunch of smaller vessels, then all those smaller vessels, the total net flow must be equal to the flow of that larger vessel. The same volume per unit time must flow through it. And if the total cross-sectional area of all those small vessels is bigger, that means the flow will be slower. Slower flow through those small vessels, that is going to mean that you have more time for diffusion to occur. It will take less time for diffusion to occur through all of this surface area, and you will have more time to allow it to occur. So this does those two things for you. More time to allow for diffusion, and more surface area for diffusion to occur across. That is why fractal branching is an important system and an important structural feature in so many biological systems. It's for diffusion. It helps with the diffusion of substances throughout a creature. Now, as far as gas is concerned, plants have flattened leaves with pores called stomata to allow for gas exchange. The stems of plants and the roots and whatnot, they have pores called lenticels. They are a porous tissue. Those allow for gas exchange. So plants don't really need a way to deliver oxygen dissolved in fluid to their tissues. Largely, they are uniformly, gas is distributed uniformly throughout the plant. In animals, you have a very complex system for gas exchange. And there's a lot of different organs that play a role, like the pulmonary system, the cardiac system, the vascular system. You really have to have a lot of different organs in place for animals. If you try to do the same thing for animals as you do for plants, having a whole bunch of holes in animals, that's illegal. You shouldn't do that, and you should probably seek help. Right. So let's talk about rate of diffusion. Diffusion, as we discussed before, is the movement of molecules from an area of high concentration to an area of low concentration. So the rate of a thing is the change of a thing over time. So if we think of those individual words, how can we calculate the rate of diffusion? What equipment should we use? You could pause the video and think about it for a second. Okay, so the equipment that you would use a ruler. You would measure the distance that thing has traveled, and you would also use a stopwatch. And I don't have the prop for that. We'll add it in post, or not. All right, so you would use a timer and a ruler. So how long did it take, and how far did it travel? Now, how long did it take and how far did it travel? Calculating that, you can determine the change over time, change in distance. You take the initial distance, you take the starting. This is your distance 1. And then if you're measuring diffusion over time, that would be your distance 2, and your time total would be 15 minutes. So if you put a dye in a well, D-Y-E, in a well, and in an agar gel, and then you allowed it to diffuse for 15 minutes, your T1, your T initial, is equal to 0 minutes. And your T final is equal to 15 minutes. If your D1 versus your D2, let's say that is, oh look, it's 120 again. D2 minus D1, your final minus your initial, is the change. Your delta D is your change in distance, is equal to 120 millimeters. All right. Your rate over time, your rate is your distance over time. So that's 120 millimeters in 15 minutes. Your final minus your initial, 15 minutes. So 120 millimeters in 15 minutes, that would be the video.